Hello and welcome to the microbial world where we learn the fundamentals of microbiology. In this session, we will be learning about the dynamics of microbial growth.
Microbial growth is a fundamental biological process describing how microorganisms increase in cell mass and cell number. It involves the biosynthesis of cellular components such as proteins, lipids, and cell wall materials, culminating in cell division and population increase. Cell growth refers to an increase in the mass or size of an individual cell as it accumulates cellular material. Population growth refers to an increase in the total number of cells in a microbial culture or population, primarily through cell. Mechanisms of reproduction. Binary fission is a simple yet highly coordinated process that ensures accurate DNA replication and distribution to daughter cells. It consists of the following stages. 1. DNA replication. The bacterial chromosome is duplicated beginning at a specific site called the origin of replication. Cell elongation. The cell increases in length as the cytoplasmic content doubles. Septum formation, cytokinesis. The plasma membrane invaginates, and the cell wall begins to form a partition or septum. The protein FTs forms a contractile ring at the division site, guiding septum formation. 4. Cell separation. The septum is completed, dividing the cytoplasm. The two daughter cells may separate completely or remain attached in chains, e.g. streptococcus, or clusters, e.g. staphylococcus. Other reproductive strategies.
Although binary fission predominates among prokaryotes, other mechanisms exist: budding, unequal division where a new cell, bud, develops from a parent cell, as in Hyphomicrobium and Saccharomyces cerevisiae, yeast; fragmentation, filamentous bacteria, e.g., Streptomyces, or cyanobacteria break into fragments that grow into new individuals; spore formation, some bacteria, e.g., Streptomyces, bacillus form reproductive or survival spores under adverse conditions. Measuring microbial growth. Because microorganisms are microscopic, direct observation of growth is difficult. Scientists measure growth by estimating cell number, cell mass, or activity. Methods are broadly categorized into direct and indirect. Direct methods: counting cells. 1. Direct microscopic count. A small volume of culture is placed on a Petroff-Hauser counting chamber or hemocytometer. Cells are counted under a microscope.
The total number of cells, both living and dead, per milliliter is calculated. Viable cell count plate count method. Only living cells capable of forming colonies are counted. Sample is serially diluted and plated by spread plate method. Pour plate method.
After incubation, colonies are counted and expressed as colony-forming units, CFU/ml. This method gives a reliable measure of viable, living, cells. Kindly study this example. Indirect methods, measuring biomass or metabolic activity. 1. Turbidimetry, optical density measurement. A spectrophotometer measures light scattering at 600 nanometers. As cell number increases, turbidity, cloudiness, rises. Optical density, OD, is directly proportional to cell mass. Fast and non-destructive, but measures both live and dead cells. Dry weight determination, used for fungi and filamentous organisms. Cells are filtered, dried, and weighed. Provides accurate measurement of total biomass, measurement of cell components or activity, quantifying protein, DNA, or ATP content. Measuring metabolic activity. The microbial growth curve. Batch culture dynamics. Batch culture is a closed system with limited nutrients and no waste removal. When growth is monitored over time, a characteristic growth curve is obtained with 4 distinct phases. Phase 1, lag phase. Period immediately after inoculation when there is little or no increase in cell number. Cellular activity. Cells adapt to the new environment. Enzymes and cofactors necessary for metabolism are synthesized. Damaged cells repair membranes and DNA. Duration factors. Physiological condition of inoculum, composition of the new medium, temperature and pH conditions. Phase 2, exponential, log phase. Characteristics: cells divide at a constant and maximal rate, population doubles at regular intervals. Exponential growth. Cell components, DNA, RNA, enzymes. Synthesized proportionally, balanced growth. Biological significance: cells are most uniform and physiologically active.
Ideal phase for studying enzyme activity, genetics, and antibiotic susceptibility. Cells are most vulnerable to antibiotics and disinfectants. Phase ill. Stationary phase. The growth rate equals the death rate. Net population growth is 0. Causes: nutrient depletion, carbon, nitrogen. Accumulation of toxic metabolic byproducts, for example, organic acids. Reduced oxygen availability, cellular adaptations, cells decrease in size and metabolic rate. Synthesis of stress response proteins and secondary metabolites, for example, antibiotics. Sporulation may occur in Bacillus and Clostridium. Some cells enter a dormant viable but non-culturable VB&C state. Phase 4. Death decline phase. The number of viable cells decreases exponentially. Causes: exhaustion of essential nutrients, accumulation of toxic substances.
Irreversible damage to cellular membranes and enzymes. Observation: population decline may follow an exponential pattern. Some cells survive longer by forming spores or entering dormancy. Here's bacterial growth curve. Quantitative growth kinetics. Kindly study exponential growth mathematics. Continuous culture systems, chemostat/turbidostat. Explain the principle of an open system where fresh medium is continuously added and used medium, with cells, is removed. Allows maintenance of the culture in the exponential phase at a desired, constant growth rate for extended periods. Significance: essential for industrial microbiology and research requiring a constant supply of physiologically uniform cells. Microorganisms grow only under specific physical and chemical conditions that match their physiological adaptations. Temperature affects enzyme activity, membrane fluidity, and nutrient transport. Cardinal temperatures: minimum, lowest temperature supporting growth; optimum, temperature at which growth rate is maximal; maximum, highest temperature supporting growth. Classification: group, optimum, degree C. Example: psychrophiles, 0 to 20, Pseudomonas fluorescens; psychrotrophs, 0 to 30, food spoilage bacteria; mesophiles, 20 to 45, most pathogens, e.g., coli; thermophiles, 45 to 80, Bacillus stearothermophilus; hyperthemophiles, greater than 80, Sulfolobus, archaea. pH affects protein structure and membrane integrity. Acidophiles thrive at pH 6, Thiobacillus ferrooxidans; neutrophils, optimum pH 6 to 8, most bacteria; alkalophiles grow at pH 8, Bacillus alkalophilus. Oxygen concentration, oxygen availability determines the type of metabolism. Classification: growth characteristics. Example: obligate aerobes require oxygen; Pseudomonas aeruginosa; facultative anaerobes grow with/without oxygen; E. coli. Obligate anaerobes killed by oxygen; Clostridium botulinum, aerotolerant anaerobes, ignore oxygen; Lactobacillus, microaerophiles, require low oxygen, 2 to 10%; Helicobacter pylori, water activity, AX, and osmotic pressure, water activity, AX ratio of vapor pressure of solution to pure water, 0 to 1. Low AX values, due to salts/sugars, limit microbial growth. Halophiles require salt, e.g., Halobacterium; osmophiles tolerate high sugar, e.g., yeasts and jams. Nutritional requirements: macronutrients, carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, magnesium, calcium, iron; micronutrients: manganese, zinc, cobalt, copper; growth factors: vitamins, amino acids, nucleotides.
Practical application of growth dynamics, food microbiology. Predicting spoilage and controlling pathogen growth through temperature control, pH adjustment, low water activity, industrial and biotechnological applications. Maintaining exponential phase in chemostats improves yield of antibiotics, for example penicillin. enzymes and amino acids, biofuels, and organic acids. Medical microbiology.